All right, good afternoon. I'm just going to transition right away to our next session. My name is Jay Shin. I'm a group leader at Genome Institute of Singapore. This is my seventh time coming to HCA Asia Meeting. It's thanks Partha and the rest of the local committee for organizing this wonderful meeting here today. The next session is kind of the tradition of Human Cell Atlas Asia, which is not really scientific, but it's a way for our community to come together. It's an opportunity for new collaborations and new ideas to formulate. The analogy that I could give is that the second meeting that we had in the Human Cell Atlas Asia in Asia, in Korea, Sham, Woong-Yang, and myself, and a few others decided, hey, why don't we come together and start doing some single cell analysis on PBMCs? And thus came the AIDA project, where we were able to formulate a grant proposal and started a whole new consortium, and we were extending this to various other countries for the last 5 years, and it's been extremely successful. So the question is, can we have another AIDA-like project coming out after this meeting today? And I hope that through the next few presentations that you'll be inspired, but also have some ideas and curiosity to engage with the speakers and perhaps start formulating ways to write your own grant proposals to initiate new collaborations. So I hope really the desire is that we'll have a new AIDA-like projects at the end of today and tomorrow, and I really encourage you to participate after their presentation, where we'll have breakout sessions and to engage with the community and the speakers. So with that, I would like to invite our first speaker, Woong-Yang Park. Just want to give you, there are three flagship projects or ideas that we have. Of course, it's not limited to these three, but these three have been mentioned quite a bit in the last few years, and we think this will be a great opportunity to create a new proposal. One is related to pan-cancer related topic. Second is around infectious disease. And third is around microbiome and their interaction with the single cells. So I hope these three themes could be kind of further investigated and hopefully initiate into something very fruitful. So with that, I'll give the podium to Woong-Yang, please.
Okay. Thank you, Jay. As Jay introduced, actually, we start discussion in the some collaboration flagship project in Jeju Island in 2018. And finally, that 5 years later, we can have some nice result and also some publication, also some asset as our HCA Asia. I think maybe it's a good time to discuss another flagship project. Last year in Bangkok, we introduced some activity for some collaboration for the cancer atlas for Asian population. So as a legacy, also maybe some as a successful story from the AIDA. I think about that maybe can leverage that information to cancer immune, right? So I think I will work with the many people in Australia and Japan and Thai. So actually, I would like to just the members of current members for Asian Cancer Atlas, but actually I hope to invite many more from the audiences. And then Natini, Janawas from Waradon University Hospital, and also Takaseki University of Tokyo, and Ankur Sharma. He is now in Perth, but actually and is now attending today, and yeah. So for aim for this project, be to understand the Asian immune diversity, and the cancer immune diversity. So we understand a lot of things related to some huge or some sustainable, some comparable difference in the Asian population compared to the other population too. So as you know, is that the immune response to the immunotherapy or other cancer response, cancer development is different from the Caucasian population. So definitely there should be some difference. So we need to understand. So basically, out of the AIDA data, probably can understand more about that cancer immune diversity. So and second aim is I want to put up the some immune states. With the immunotherapy. So not many cases, but actually we can include that kind of sample, because most of cases we will analyze the cancer patient data. So definitely in some cases we have some sample we treated with some specific treatment. If then we can understand more about that, some effect of the different drugs in the different populations. And third one will be definitely we need to develop this the database. Also, we need to develop some platform to collaborate with the other members in HCA Asia, also other scientists in Asian continents. So to achieve this kind of aims, actually our ACA project be covering collecting the data for single-cell multiome data, including the single cell, also in the spatial and other multiomic data together. And we'll operate that database to so many different databases, but actually we'll also include the database to maximize the utility of those data sets. And third one will be we will try to analyze the immune diversity, also many the techniques developed by our members. Probably if then we can understand more about the cancer immune diversity based on different methods, the single-cell HCA or other AI-based methods. So I will introduce some activity. Also, it's not the initial pilot stage. So I think maybe we can develop more idea and then more. It's not formulated well. So I think maybe individual effort by Anku and Yutaka and myself. So I want to speak briefly about that. So Anku actually he collected liver cancer samples from initially he's starting from the Singapore, but also including some samples from Australia or other countries too. So basically we will start with the PBMC of cancer patient plus the spatial data for the cancer patient. As you probably he submitted a paper in the Nature Cancer, and actually we see some difference in the different population, different stage. So in the PBMC, also here to see the some niche, leukocytal niche distribution is different from responder and responder based on some immunotherapy response. I think maybe definitely the same for other cancer types, or maybe same for the other population. Also, as Shyam presented today, there's BBs. For example, in that I don't see why with Treg in Korean population is low, but actually definitely it will related to some cancer, immune response in the Korean population too. Also, Natini, she also operated some activity for single cell sequencing plus in the spatial. So she collected samples and the clinical data together in Thai. So based upon last thing that we present something in the Bangkok, and then follow-up meeting in Busan on this year in the May, and then we discussed what you can do, and then so maybe mostly that we are decided to focus on major cancer type first in the initial stage, and also to make a sustainable structure. Actually, we would like to find some funding from outside. But as you know, it's the same situation for other flagship project or even for IDA. Actually, the beginning actually is not easy to make some nice the plan for the to operating those large data sets. So we'll keep trying to apply this any international funding, but the beginning actually we will start with the Japan, Korea, or maybe other national funding inside, so to support that activity. Also, the time is changed. Also, 5 years before, actually, we started with the single cell PBMC, but now in the year for the spatial biology. So we're going to include that spatial technique for this project. So there's three main cancer types. Lung cancer initiated by Yutaka in University of Tokyo, and HCC led by Ankur Sharma in Australia. And this colorectal cancer is by myself. We plan to collect 100 spatial data each, and then from based on their own the some funding or something like that. So, and then we will integrate them together. So we do not have any single protocol for that, how to collect the samples, but actually we should consider the numbers. Also, we decided to start with a xenium. Actually, 500 gene panel by each cancer types. Also, we would like to put in the 4 samples in the one slide, so maybe demand will be 1 by 0.5 cm. And then, now it's starting to produce data, so I think once we have the data, probably can discuss the other steps like the QC or analysis, something like that. Also, in parallel, we will also need using public or private data for the Visium to compare the Caucasian versus Asian population samples. And myself and I have about 50 or 60 Visium data for 5 different cancer types. So I think maybe we can start with the comparison with to find any Asian specific pattern, right? So I think maybe so briefly we already did some xenium study for the HCC by Ankur, and then he found some interesting features by their own samples, and also we do experiment with the colon cancer with biopsy samples, and then we do see some specific features. So I think maybe that is platform currently we running for liver cancer, also in the colorectal cancer. So in the flagship, breakout session, actually we will include a discussion about these topics, like the how we can in the IDA. Actually, I benchmark a lot for the IDA, but actually it's not exactly the same situation. So I think maybe we need to discuss how we can make some sustainable structure for the ACA. Also, how we can standardize the data. Also, how we can, mostly in the both spatial data. And also, we should make a database platform, but actually who and how we can make those database platform. Also, happy to collaborate all the members, but actually how we can decide that the collaboration scale, something like this. So I'd like to discuss about these topics in the breakout sessions. Okay, thank you.
Thank you, Woong-Yang. Maybe if you can, maybe I'll take one question from the audience. Jump.
It's great to see Genomics Consortium forming. Hopefully, it's a landscape that can be globally in this kind of effort. But I guess one big thing to tackle, which you alluded to in your slides, is how do you put the data together and share it? I think that part is very new for specialists.
Right, so that is one thing we should consider, but actually, for example, in the colorectal cancer, actually, I will run my CRC project in 100 cases myself, and I also welcome to receive the sample from outside. So as you know, for xenium, actually, we need the paraffin block. So if I can have received them, so we will discuss our breakout session, how we can handle the samples together. So I think it's currently the plan be a centralized experiment in one center. Yes, yes, yeah. So single cell, PBMC sample shipping or some data generation is not easy at the time. So I think maybe we can, spatial is much better to the logistics for the samples, right?
Great, thank you. Okay, let's invite the next speaker. And again, if you have further questions or comments or suggestions or ideas, these will be followed up with a breakout session after this. So feel free to engage with Woong-Yang and contribute your thoughts as well. So next we'll invite Yukinori Okada, who's a PI, a team leader at RIKEN, as well as a professor at University of Tokyo. And virtually we'll have Ponpan from Thailand joining us. And they will talk about the flagship project involving infectious disease. So please.
Okay, hello everyone, this is Yukinori from Japan. Really sorry for my trouble for uploading the slides. There was some contamination, but okay, let's start our flagship proposal for the infectious disease. So this session, I'm Yukinori from Japan, and hey, come on, Waradon.
Oh, I'm Waradon Sungnak. So Ponpan has asked me to join with a flagship proposal as well. So I will be talking a little bit at the end.
Okay, so let's start the discussion. So yes, there were several points to discuss. Of course, details we are going to have the time to discuss in the COVID breakout session. So anyway, we want to make some good network to make the infectious disease genomics network around this HCA Asia realm. So we have some short-term goal to, okay, which infectious disease we should prioritize, which kind of tissue or single-cell omics analysis we should do, and our methodology. And we also need to have which level of human genomes or host genomes, and how we should develop tool integrate post-pathology analysis to explore system responses. So there are many things we should discuss to how to make these networks. But I think because, yes, these Asian countries the focusing good topics for the infectious disease. So making a collaboration network through this community through the infectious disease single-cell omics is a good target for us. And of course, the mid-term goal is to construct pan-Asian network and practical pipeline to elucidate infectious disease biology and make the catalog within Asian diversity of infectious disease omics. So infectious disease is not same even within the Asian countries. We should definitely want a catalog at this point. So actually we can discuss details, especially which disease we should focus on at the upcoming breakout session. But here I would like to introduce several of the candidate disease which we have discussed earlier. So we are going to talk on 3 diseases. And okay, first is the NTM. I would like to introduce Dr. Namkoong who is mostly leading this NTM genomics throughout the Asian countries. So Namkoong, please go ahead with your presentation, and I can maybe help you to proceed the slide.
Okay, thank you, Yuki. Thank you, Yuki. My name is Namkoong. Sound is okay. So sound is okay.
Okay, please go ahead.
Okay. So my name is I'm a IT patient in Japan and Korean. Okay.
Namkoong, sorry, the sound is difficult to hear in this room due to some technical reasons. So maybe I can instead do some brief presentation on this. So NTM is a non-tuberculosis microbacteria, and there are so many species. But I think yes, this is really a severe disease, especially in Asian countries. And because there are so many diversity in NTM species, we definitely need to proceed this analysis. And yes, there's so many environmental factors affecting these diseases. And yes, NTM is relatively hidden in the TB, but these are different diseases. We should definitely attack both these diseases. So, because to dig out the patient from misdiagnosis from the TB, we should definitely proceed NTM genetics and genomics throughout the agents. And Dr. Namkoong is a leading researcher on this disease. He already has some GWAS or multi-country or multi-Asian countries. So he should definitely have the host genetics insight of these diseases. But and yes. This is what he's going on. Actually, he's going to many NTM pan-GWAS of Asian countries, but he also massively conducting a single-cell analysis of the infectious patients or some stimulation conditions. So actually, we want to expand this NTM genomics and single cells throughout Asia through different strains, host-genetic pathogen interaction. So I hope some of you have strong interest on this disease too. Okay, I'm sorry. I have to emphasize again. Here's the strengths of infectious disease research in Asia. So there are so many unique diseases. So dengue, malaria, And also there are so many, unique response from the European countries like COVID-19 and TB anyway. So, but they are heterogeneous, so we should definitely proceed such omics analysis. Okay, thank you, Namkoong. So I've just have brief presentation instead of you, but we're really happy to discuss in the regular sessions. And I think, this is an overview slide.
So I think Ponpan will
Okay,
take
Ponpan is going to take it. Ponpan, please go ahead with your presentation.
Hi, can you hear me okay? Hi, can you hear me okay? I think there's lots of echo maybe.
Ponpan, again,
I think there's lots of echo maybe.
Sorry, Ponpan, maybe the sound is difficult to hear in this session, some technical. So Waradon is going to take over your presentation, sorry. Yeah.
So I can help Ponpan with that. So we just mentioned that the strengths of applying the single-cell technology and all other related technology in infectious disease in the Asian population. But there are also challenges in kind of establishing a sustainable network for this kind of research due to disease heterogeneity. So diseases in countries like Korea or Japan in temperate climate would be different from diseases in tropical countries, right, like India or in Thailand. So like the selection or the disease would be an issue, right, which one we should emphasize on. There are also legal barriers, so different countries would have different laws, so in terms of transferring the clinical samples, especially in the context of infected samples, that would be another challenge that we need to overcome. There are also the issue of balancing contributions. So to some extent, there could be a clinical sample from more developing countries. So to what extent we should do to facilitate the contribution in developing countries that in the end scientists in developing countries would not just end up being sample contributors, but more of the more intellectual or technical capability would be upskill as well. And there's also a lack of standardization in protocols and methodology. So scientists in different countries, in different locations, we sample in different ways, and the processing and the analysis are different. So could there be a way that we could have a unified framework or a consensus framework such that in the future, when we want to integrate everything together, things will be more practical? And also a lack of effective communication channels. So, if we can improve on that, like as in the case of HCA Asia, then that would promote the collaboration better as well. And there's also issues of political and geographical sensitivities. Of course, to some extent it's obvious, but at the same time, this could be a challenge in terms of long-term collaboration. So one direction or one thing that, like we discussed, maybe we could start with something like a framework-centric project where it's just developing a common framework that can be universally applied to various infectious diseases. Like from the host to host pathogen interaction to the pathogen identity themselves. And the project can be something horizontal, so not just, like focusing as a unified, like single project, but more on any like infectious diseases, keeping the independence and flexibility to pursue separate research projects while fostering a shared framework for core methodologies or experience exchange. And as you can see in some of the talks mentioned today, right, so like great talk from Jinmil, so we could see example of some application on the extension toward the pathogen and host pathogen interaction. So far, we focus on the host side. We understand human cell atlas, we understand single-cell transcriptomic phenotype of the cell, and then extending toward the multimodality, better understanding host genetics, and then like more on the other layers from the genetics and the phenotype. And then there's a whole universe of microbiologists who study pathogen microbial genomics, and that's a separate universe. And there's some degree of people, like us, working on post-pathogen interaction, we find some correlation on cross-sectional, longitudinal aspect, or biology or infected cell, like example from Jinmil talks earlier on, and then TCR, BCR repertoires, or antigen-specific BCR and TCR. So could we have a framework that, like once we collect a clinical sample, then we could get the most out of the clinical sample that we collected, and how we would be able to, like convince microbiologists in, let's say, like in a university in Thailand that they would let us, like they would analyze the microbe site and we analyze the host site. So how we talk and have a framework for all the stakeholders in such a way that everything would be a smooth for a long-term collaborative network. So something like, like potential steps to implement. So, like maybe we should gather all the experts, right? Like not just people who work on the host site, but maybe microbiologists, bioinformaticians. So, like what we actually need in order to establish a shared framework, identify core elements, like something that could be common across different infectious diseases. And maybe we could start with that and, like start it out with some we could start it out with some pilot, like few selected, infectious diseases, and then see how we could refine that, and then we might be able to extend toward other infectious diseases as well.
Okay, thank you, Ren. So maybe this is our presentation. So in the breakout session, we are welcome for your interests and opinion and votes for the diseases. And actually, we should launch some single-cell collaboration analysis for some targeted infectious diseases. Thank you for your listening.
Thank you. So for the interest of time, we'll move on to our third speaker of the session, Archita, who will then talk about the microbiome. So relatedly, we just heard about infectious disease and viral to immune host interactions. Perhaps bacterial to host interaction could also be of interest, and an opportunity to build a reference map or the framework to work together on this. I just want to, while Archita is getting ready, just want to mention that there are 2 breakout sessions, meaning these 3 themes will be running in parallel for one session. And so you can choose one of the 3. But if you really want to, if you're curious about other sessions, then you can join the other breakout rooms in the second session. So you can choose, you can decide to choose one and stay there 2 sessions, or switch between the 2 sessions. So with that, I'll give the podium back to Archita, please.
Thank you, Jay. I hope I'm audible. My name is Archita, and I'm a group leader in Australia, Western Australia. And my lab is basically interested in microbiome and its impact on immune system. But today I'm going to talk about why microbiome and why in HCA in totality, not just HCA Asia, we should have included microbiome yesterday. So we know that human bodies are constantly evolving ecosystem, and if you really see, every organ in human body is like an organism in itself, and it is an entire ecosystem in itself. There's a lot of microbes that live in every organ of our body. And if you have to collectively count the amount of genes these microbiome encode, it will be 100 times more than what our body encodes. So it's basically, if you really see from a gene standpoint, we are more microbial than human. And a lot of functionality and metabolism and immune system regulation in our body is interlinked with microbiome. So it is very important to study human cells or human organs or human functioning in conjugation with its microbial mates. And it becomes more important when we are talking about immune system, because immune system is all about protecting us from what's outside or what is foreign. And microbiome is the first layer of barrier and first layer of protection that comes into picture and gives us the natural defenses that's present in our body. And this microbiome sort of trains our immunity to know what is good and what is bad. So it just doesn't start killing everything left, right, center. And this is why microbiome and immune system together, in conjugation, is the way to understand immunity in humans and in any animal forms, to be honest. In the past, we have shown that this microbiome, this association of the microbiome with the immune system, it starts quite early in human life, especially during gestation, during second trimester of gestation. There are microbiome, like there's microbes present in our body, in mother, which gets transferred from the mother to the fetus in different organs of the fetus, and it sort of trains the fetal immunity in a way that it retains a memory for these microbes. And it's not just us, but several other studies in the past decade have actually shown the impact of microbes and microbial products, metabolites and antibodies which are in response to microbes. They help to shape the immune diversity in the fetus. And that's why it becomes important to study this, because that's the start of immunity. And that's where the immune system is getting educated to face what's going to come outside later when the baby is born. And this is in a way kind of natural immunization, which scientists have been believing since 100 of years, that what happens during gestation and right after that is sort of providing natural immunization to the baby. Now, what we are doing currently in the world is basically trying to also understand the natural immunization, but also trying to boost the immunization process, and that is what the vaccination comes into picture. So when a baby is born, there are myriad of vaccination, immunization schedules, which is given to the baby in order to fight the microbes that might come later, even though they are not exposed at present. But in a way, if you really see, these two are not mutually distinct entities. They are quite intertwined. So we have certain level of natural immunization, natural immunity from the mother and from the environment, and then we combine it with the vaccination, and there we are, protected, nice and happy. But like Shyam mentioned earlier, disparity is something which holds us all together to study these kind of systems. And in this system particularly, where there is a baby involved who is, this is the most vulnerable population in our world, there's a huge disparity across world in the response of these babies towards vaccination, especially if we have to divide the world between low income and high income settings, or the settings where there is a lot of migration going on, or different food habits. There's a huge disparity here, and we still don't understand how these vaccines work, but we are still giving the same kind of vaccination without any booster, without any adjuvant to all these babies, and we don't see the response in all the time. So this is a time to understand what's happening, what can we do to enhance this process in a way that the babies are more protected in areas where they are actually less responding. So the idea here is, in this whole microbiome diversity atlas, to study the microbiome of the newborn, and this is where I come into picture because I'm interested, my lab is interested in newborn immunity and microbiome, but in a way it's open to all sorts of ages and all sorts of settings. But we can start with the microbiome in newborn, how it develops across time, during gestation and then right after birth, and how it impacts the vaccination response in early life. So here, the kind of sample and the kind of assays that we'll be doing is we'll collect stool samples and we'll do metagenomics, we'll do PacBio, because by that we can sequence the entire 16S ribosomal RNA and get to know the diversity up to species level. And we will also collect some blood to study the antibody titer of the response to certain vaccines, and also the neutralization ability in our immune system. So the vaccine that we have chosen currently, and of course it is open to the more the merrier, but right now we are focusing on hepatitis B vaccination, which is given to the babies when they are just 12 hours old. So within 12 hours of their birth, this vaccine is given to the babies. And then the idea is to collect the blood samples after a few days and look at the antibody titer and neutralization assays, and also look at their microbiome over time. And then sort of look at the correlation between, if there is a certain type of microbe associated in early life, they might have a better immune response in this case. And then we start with Hep B, and the idea is to expand to more early life vaccines. Now consider a different setting. So this is where a healthy baby comes, gets the vaccination within 12 hours of life, and is quite protected. depending upon which part of the world the baby is in. But what happens if the baby is born preterm? Irrespective of which part of the baby of the world the baby is in, if a baby is born preterm, they are always vulnerable to infections. Now, it depends upon the kind of infrastructure and the kind of hospital facilities are there in those countries, but the baby inherently is quite vulnerable to infection. And these babies have to wait for even immunization. They don't get immunization right away. So they are kept in enclosed containments, and then they get immunization only after they are healthy. So they were vulnerable to start with, and they get further vulnerable because they cannot take vaccination at such early time. So we are taking baby steps in this direction as well, and with our collaborators, we are building this cohort. So with our collaborators in Australia and Singapore, so we are building this cohort where we are trying to study the preterm and term babies' microbiome, and eventually the idea is to bring in the blood aspect as well. So what happens when a baby is born preterm? A lot of times the babies are given probiotics, and studies have shown that giving these probiotics actually increases the risk of fighting towards infection in this baby, not the risk, but basically it's protected. If the probiotics are given, the baby is protected and they don't get infected as easily as a preterm baby who didn't receive probiotic. But these are very early stages of studies and we don't understand how that happens. So we are collecting longitudinally the full term and preterm cohorts for 1 week, 4 week, 4 month and 1 year of life. And we are sort of understanding the comparison between these microbiome and the dynamic changes in these microbes. And the early data shows that, I know it's a busy slide and I don't have much time, but just focus on this one. So the first column represents the preterm babies that don't have probiotic, and the second column is the preterm babies that have been given probiotic. And these babies have certain developed eventually with time. By 4 months, they have developed certain microbiome, which is very good for the for fighting infection for the baby and also for metabolism, proper metabolism of the baby. And it basically represents something similar to what a full-term baby has. So we are kind of trying to skew the microbiome of the baby of a preterm baby towards a full-term baby. So it's more like sort of supplementing something so they can be better protected. Now the idea is if the blood response also looks something similar, if probiotics are given, or if vaccines are given along with probiotic adjuvants, which we have seen in the cases of rabies vaccination and in certain other vaccinations, certain probiotics and metabolites like retinoic acid have been given, and they have proven to be really good. So this is the plan, and then if you really focus on these babies up till, 1 year, there is actually no difference between the preterm and term baby, while there is a huge difference between the preterm babies that were not given probiotic. So I think my time is over. The idea is so right now we are starting from Asia Pacific, but the idea is to expand this and look at the global microbiome development and also across ages and across different ethnicities, different populations, and eventually just go global with this. And again, the idea still remains to understand what happens during development and how this development sort of entails the way the human being or the baby or the adult responds to infection and everything. So this is the collaborative network at present, and this could be you, and if you work on any disease, you work on any sort of developmental aspects or the disease aspects, please feel free to talk to us. We also have a microbiome breakout session after this, so please come and talk to me, and it'll be great to have some discussions around this. Yeah.
Thank you very much. So for now, we'll close this introduction session. Again, please come to and participate in the breakout sessions. And please probe the speakers, but also feel free to contribute. So with that, we'll end this, thank all the speakers, and we'll move on to our next session. Before that, Partha has some announcements to make. Yes.
Thank you, Jay, for conducting this session. Thank you all the speakers who introduced and will continue with these discussions during the breakout session. It's my pleasure to invite Professor Piero Carnenci, who graduated from the University of Trieste and then moved over to Japan for a long time in Riken, and right now both in Riken and he's heading the genomics in Human Technopole in Italy. So Piero, this is the first Funders talk, and Piero is going to chair that talk.
Thank you, Partha, and I'm just chairing, but just my big pleasure to introduce to you Dr. Yukio Kemmochi, who kindly agreed to come here and give a talk about funding. He's going to talk about exploring Asia, JRP, and SATREPS. Dr. Kemmochi got his a degree from the Yokohama National University in 1991. He has a very extensive industrial experience with, he has been working at Rompolan and a Barak corporation. Then he went back to study, and he got a PhD in pharmaceutical science in 2004. And then he joined the Science and Technology Agency, which is a very important agency in Japan, and he has been covering many positions. And just for the interest of time, probably it's better if you start his talk, and then we will continue the discussion. Please, Dr. Kemmochi.
Thank you for the introduction, and I'd like to express my gratitude to having this opportunity to introduce our activities in this Human Cell Atlas Asia Meeting. Okay, now I am Yukio Kemmochi from Japan Science and Technology Agency, JST. JST is one of the biggest public research funding agency in Japan. And today I will introduce two of our research funding to support international collaborative activities. One is Asia JLP, and the second one is SATREPS. But before going into detail, let me introduce our JST's policy on research support. I want to go into details of United Nations SDGs, but there is a similarity between SDGs and JST's research policy. As you know, the aim of SDGs is eventually to establish a world with well-being. It is the same. JST supports research activity, but supporting research activity is not our goal. Our goal is to establish or to contribute to establish a world with well-being, and research is for us as a tool or means. And as highlighted in red here, you can see that the similarity between SDGs and JST's policy, and the basic principle of our international cooperation is here, just like SDGs. Okay, now let me continue to Asia JLP. Asia JLP was initially proposed by Japan in ASEAN-related event in 2010, and then officially established as an international collaborative program in 2012. And this initiative is among East Asia Summit member countries, or EAS countries. Among 18 EAS countries, 14 participates actively to our EASIA GRP program. And 25 member organizations or funding agencies are representing from these 14 countries. And you may notice gaps or blanks here in 18 EAS member countries. These are seats, reserved seats for Brunei, China, India, or South Korea. I'm having this presentation now in Kolkata, India, but unfortunately, India at this moment is not our member organization. So we cannot accept application from India. But of course, India is eligible to join our EASIA program. And I truly wish to welcome India someday near future. And EASIA has two core activities. First one is supporting multilateral international research project. And please remember this keyword, multilateral, not bilateral. And second activity is promoting a network between among research communities in Asian region. And during the last decades, we have supported more than 90 projects and 20 events in Asian region. And this is our funding framework. As I mentioned, EASIA is a multilateral international collaboration. So to apply EASIA GRP, the researcher needs to have teams consist of 3 or more countries. And EASIA is a so-called co-funding system. Co-funding means that each PI receives its fund or support from its own country's member organizations. It means that PI from blue countries can get support only from their blue country's member organization. So, and the country of red PI can get fund from a red country's member organization. So, under this scheme, we cannot accept application outside EASIA member countries. And this is a field of cooperation. And we have 7 fields of cooperation. First one is materials, including nanotechnology. And second one is alternative energy. Alternative energy consists of both bioresources, like biomass, and inorganic chemistry, like electrochemistry. And third one is agriculture, including food. And of course, health research. And the next one is a disaster risk reduction. And these research fields include urban planning. And this one is a little bit longer. Advanced interdisciplinary research towards innovation. In short word, cutting-edge technology. And the last one is environmental matters, like climate change and marine science. And sometimes we have open code for single domain. But recent years, we have overarching research field, like climate change and health. Actually, this overarching field, climate change and health, is this year's 2023's research field. And I think this is a very good opportunity to conduct interdisciplinary research. Because under this scheme, meteorologists and health specialists have a team together. And typically we have 2 or 4 fields of cooperation for open code. And in general, we open the next year's code in December, in mid-December. So now we are preparing for press release. And I cannot tell into detail here under recording, of course. It's because before press release. But if you want to know what kind of code will be designed for 2024, please ask me after this presentation. And this is a structure of eAsia JRP, how open codes are designed. And please disregard in detail. The important thing is that initial step to create a next year's code is based on each country's member organization's proposal. It means that only a member organization or only eAsia member organization country propose a code. And if you are interested in applying eAsia JRP, please visit our website here, and we will release the next year's call information in mid-December, after 15th or hopefully 15th or maybe 12th. 12th? No, not possible. After 14th, I think. Then next, I will also introduce about SATREPS. SATREPS is another Japanese major funding program to support international collaborative research. And as I mentioned, eAsia is a multilateral collaborative scheme. But SATREPS is a bilateral collaborative scheme between Japan and developing country. And the field of research are these 4: global warming, bioresources, natural disaster, and infectious disease. And the first 3, global warming, bioresources, natural disasters, are funded on the JSD scheme. And the last one is funded by AMED, another Japanese funding agency. And that SATREPS is it's a collaborative. Collaborative means a collaboration between funding agency. A collaborative scheme between JSD, research funding, and ODA, official development assistance. So here, as I mentioned in Asia, the fund or support cannot cross the border, but in SATREPS scheme, Japanese funding agency can support foreign researchers. So if you are interested in SATREPS or Asia for the next year's applications, please feel free to ask me after this presentation. And thank you for.
Dr. Kemmochi, thank you very much for your presentation. So I think we are running out of time. So, but I would like to know if there is anyone who among the attendees who is willing to make any question for you. And while we are waiting, I would like to ask you. So, based on what you have seen at the meeting, do you see any possibility that some of the subjects discussed here could be part of the application,
yes,
if you can say?
I think that in this community, human-centered others community, you have a lot of tools for analyze in detail of human-centered activities. So I think that we can expect interdisciplinary research collaboration between this community and the EAGR community.
All right, thank you. I would like to keep it short because we are already supposed to finish the coffee break in 3 minutes. And again, Kemmochi-san, thank you very much and for your
thank
important.
And we are waiting for your application.
A cup of tea or coffee, whatever you like, and then reassemble in a few minutes, maybe after 10 minutes, in 3 different rooms. These are the 3 parallel breakout sessions. Those of you who are interested in joining the Asian Cancer Cell Atlas breakout session, please remain here or please come back here. Those of you who are interested in joining the infectious diseases breakout session, please go to the room called Sapphire, which is the one that's after this. And those who are interested in joining the microbiome diversity and human health, it's a room that's between Sapphire and this crystal room. Those are the 3 rooms. Again, there are 2, after the first breakout session, there will be a second breakout session, and you are free to move around during the second breakout session. You can go to another session if you so desire, if you don't want to remain in the session where you were during the first breakout. So you're free to move around the 3 rooms in the second breakout session. Feel free to make yourself available and discuss so that some other new projects can actually emanate from this meeting. Thank you very much. We come back in about 10 minutes.